Hi all, welcome for this session. So today we're going to learn about the mechanisms of drug absorption. So we're going to see this in two parts. So myself, Dr. Vedhamurthy Joshi, Professor Adi Chunchandgiri College of Pharmacy. So what is absorption process? So absorption is a process of movement of drug from the unchanged drug from the site of administration to the systemic circulation. So this absorption process is a complex process, so where the water soluble, water insoluble, and then wide varieties of food nutrients and then drug particles, so these are going to be absorbed. So that's why, so this is a complex process. Then many mechanisms are involved in this drug absorption process. So broadly, we can divide this or we can understand this by three basic mechanisms. The first mechanism of drug absorption is transcellular mechanism or intracellular transport. The second one is paracellular transport or intercellular transport. Then the third one is vesicular transport. So here, the first one, that is transcellular mechanism. Here we have another two types here. One is passive transport process. Then the second one is active transport process. The second one, that is paracellular mechanism or intercellular mechanism, the permeation through tight junctions of epithelial cells. Then second one is persorption. Then the third one, that is vesicular transport. Here we have pinocytosis and phagocytosis. So these are the classifications of these transports. Let us learn one by one. So this intracellular transport or transcellular mechanism. What is this? So all these mechanisms are depicted in this picture. So kindly observe it carefully. The first two that is there here. So there is transcellular mechanism. Here the drug travels from the epithelium and then enters into the cytoplasm and then leaves the cell and reaches the capillaries. Okay, so the drug is traveling through the cell cytoplasm and reaches the other part and then reaches the capillaries. So if the drug transport occurs like this, so then this is called as transcellular mechanism. So please remember this. So this involves three steps. The first one is the drug enters to the membrane and then it enters into the cytoplasm and then leaves the cell from this one through that one. And then it may reaches to the other cells or it may reaches to the capillaries, so whatever. So this transport is called as transcellular mechanism. Then the second one is paracellular mechanism. In this paracellular mechanism, the drug used to travel between the junctions of the cells. So when you consider that there are two the epithelial cells, so these epithelial cells individually, so they are separate and they are together with a tight junction. So this tight junction will vary, okay, depending upon the epithelium actually. Okay, so in this epithelium, the tight junctions, if the drug is transported, so if the molecules are very small, so then these used to transport through this paracellular mechanism. So that is paracellular mechanism. The paracellular mechanism, the drug transport is through the tight junctions that are available. Then the third one is vesicular transport. In this vesicular transport, so we have the contents, that is the drug. If the drug is there, so it reaches the cell membrane and then the cell membrane itself folds into the vesicle and then it travels into the cytoplasm and then this vesicle reaches the other part of the cell, then it releases the drug that it had hold and then it releases into the capillaries. So if the drug transport done like this, so then this is called as vesicular transport. So this vesicular transport is of two types, as I told you. If the material is solid, so then we call it as phagocytosis. If the material is liquid, this is called as pinocytosis. So that's why actually this is called as the cell eating and then cell drinking. So let me revise this. So the transcellular mechanism, the drug travels from the cell and then reaches the cytoplasm and then reaches the other part of the capillary, other part of the membrane and then releases out. So if the drug travels through the cell, so this is called as transcellular. If the drug travels across the tight junctions of the cell or epithelial cells, so this is called as paracellular. Then the third one, that is, so if the drug travels without by folding the membrane and then traveling across the cell cytoplasm without coming in contact with the cytoplasm, so this is called as the vesicular transport. So there are three mechanisms that are involved in this one. So the first one, that is the transcellular mechanism, involves the passive diffusion. The second one is pore transport. Then the third one is ion pair transport. Then the fourth one is facilitated or mediated diffusion. Let us see these in brief. Passive diffusion. Passive diffusion is also called as non-ionic diffusion. More than 90% of the drugs are absorbing through this diffusion. The basic driving force for this diffusion is electrochemical gradient. What does it mean? So it is nothing but it works on the principle of diffusion, that is Fick's first law of diffusion. So what does the Fick's first law says? So Fick's first law says that, or states that, the drug molecules diffuse from a region of higher concentration to a region of lower concentration till the equilibrium is attained. So this is the formula for this Fick's first law. So that is dq by dt is nothing but a small change, that is the small amount of the drug that is transported with a small change in the time is directly proportional to Cgit minus C. So what is this Cgit minus C? So this Cgit minus C is nothing but the concentration gradient. So this concentration gradient is the driving force. So and it is also dependent on the D, that is diffusion coefficient, A, that is surface area, and then K, so that is Km by W, that is nothing but the partition coefficient of the drug molecule. So briefly, the partition coefficient is nothing but the ability of the drug molecule to partition when the option is given in the oil or water. So if the drug loves to be in oil, so then the partition coefficient of the drug molecule is higher. If the drug loves to be in water, so then the partition coefficient is lesser. So the lesser the partition coefficient, dq by dt will be lesser. So the higher the absorption area, that is dq by dt will be more. The diffusion coefficient, that is d, is higher, so then the diffusion is also higher. So till this diffusion process will continue till the Cgit minus C is always there. So Cgit minus C is equal to 0 means, say, the concentration that is available in the GI tract and the concentration available in the blood plasma. So if both are same, so then the diffusion will cease. Ideally, it doesn't happen. So because of which, so it is called as sink condition. So this is about the passive diffusion process. Next one is pore transport. So this pore transport is also called as convective transport or bulk flow or filtration. So bulk flow or filtration. So please remember this. So the driving force for this one is hydrostatic pressure or the osmotic pressure differences across the membranes. So the process is very much important in the absorption of low molecular weight substances. So the lesser the molecular weight, that is less than 100 Daltons, so then the drugs or the substances will take this pore transport. Okay, so these pores, the pores actually they are very small, and then if the drug particle is still smaller than the pore, so then the drug will be filtered. So that's why, so this process is also called as filtration. And whenever the blood flow is there, so through that blood flow, so the drug is traveling through the small pores. So that's why it is also called as the bulk flow or the pore transport. So example for this one is urea, water and sugars. So these will transport with the help of like this. So then chain-like substances or linear molecule substances can also follow this pore transport. Then drug permeation through water-filled channels is also, we can see this poor transport in case of cerebrospinal fluid, the filtrations are the kidney excretions, renal excretions. So all these, so the poor transports, we can see that. Next one is ion pair transport. As the name indicates, so there is an ion or so there are two types of ions. One is positive, that is cations, and then negative, that is anions. So if cations are there and then there is a pair that is available or existing inside the cell, so that is ion and pair. If the molecule is cation and then there will be a pair, that is anion. So both cation and anion club together or interact together, or actually they are not bind together, so to form an ion pair. So this ion pair is neutral in nature. So this ion pair has an ability to transport or to travel from the cell membrane. So because of which, so the ion pair transport, we can see that. So it is example for this one is, so there are many drugs, so that is the propranolol basic drugs. So these will travel through this one, and then quaternary ammonium salts, as the name indicates, benzalkonium chloride, then benzethenium chloride. So these are the cationic absorption process, cationic molecules. So these will be absorbing through this ion pair transport. So the example for this one is here, the diagram explains the same thing. That is a cationic drug. If it is there, then there is an endogenous anion that is available. So both club together to form a complex, that is neutral ion pair complex. So this ion pair complex has an ability to travel through the cell membrane. So it is traveling through passive diffusion. Once it reaches inside the plasma, so then it again divides into two. So where the drug will be separated and then the endogenous anionic complex will be separated. Some polar drug molecules like monosaccharides, amino acids, vitamins, so these will be absorbed much faster and without the interference of this concentration gradient. How do they absorb? So they absorb through this carrier mediated transport. The mechanism involved is the carrier that binds reversibly or non-covalently with the solute molecules to be transported. So what happens here? So the drug molecules will be binding to the carrier non-covalently, means a loose bond that is there and which is responsible to transport from this region. So this carrier solute complex traverses across the cell membrane to the other side where it dissociates and discharges the solute molecule. So the carrier then returns to the original site to the complete the cycle by accepting the fresh molecule of the solute. So the carrier membrane or the carrier membrane proteins, so they are limited. There are certain drug molecules like monosaccharides, amino acids, vitamins. So these will be absorbed without the rule of this concentration gradient as we predicted earlier. And their partition coefficient values are also poor, but still they are absorbing much more faster. How do they? So they follow this carrier mediated transport. In this carrier mediated transport, the mechanism involved is the carrier is the one so that binds reversibly or non-covalently with the solute molecules to be transported, and this carrier solute complex traverses across the membrane to the other side where it dissociates and discharges the molecule. Here what happens? So the drug molecules will be binding to the carrier, and then this carrier is available on the membrane, and then this membrane will return or traverse across the membrane, whether it may be outside, whether it may be inside. So the drug will be transported from one region to the other region without the interference of this concentration gradient with the help of these carriers. So where the drug will be transported by binding to the carrier covalently, non-covalently or loosely binding. So these will be transported and then again the carrier will return to its original position. So this is carrier mediated transport. So here you see this. So when we see the absorption of passive diffusion and then carrier mediated transport, so you can see that the rate of drug absorption process and then concentration gradient. So here as the concentration increases, the passive diffusion goes to a straight line. Means as the concentration increases, the rate of absorption is also increasing. So it indicates that the passive diffusion follows first order kinetics. And then when we see this carrier mediated transport, initially the rate of absorption will be much more faster, then later the drug transport will cease or the transport will be coming to the maximum limit. So later that the rate of drug absorption process will be limited. So this process of the first order kinetics initial, then later zero order kinetics, so this is called as carrier mediated transport or mixed kinetics. So the carrier mediated transport is found in mixed rate of kinetics. So next one is facilitated diffusion. So it is the carrier mediated transport system that operates down the concentration, that is downhill process. So where the carriers are available but it is coming through the concentration gradient only. Means higher concentration to lower concentration the drug will be traveling. Okay, the driving force is the concentration gradient, so the energy expenditure is not involved. So please note that the energy expenditure is in terms of ATPs. So here you can see the diagram here. So next one is active transport process. So this active transport process is also a carrier mediated transport. So here what happens? So the energy is involved in this. So why the energy is involved in this? So because the transport is not from the higher concentration to lower concentration following concentration gradient, but it is the opposite, where the molecules are traveling from the lower concentration to a higher concentration. Okay, so for this region the energy is required, so the energy is utilized in forms of ATPs or NADPs. Okay, so there are again two types of this one. So there is primary active transport, then secondary active transport. So again we have in the secondary active transport we have symport and then antiport. So symport means so two molecules will be coming and they are entering on the same side. Then counter or antiport is the one, so where the one molecule gets inside the cell and then the other molecule or equivalent charge of that molecule will be transporting away or in the outside of the cell. So that is antiport. Thank you.